Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is July 18, 2025. This video is on a Nature article titled Imidazole propionate is a driver and therapeutic target in atherosclerosis, published online on 16 July 2025, open access. The associated content is a News and Views article titled Gut Bacteria Make a Molecule That Promotes Plaque Buildup in Arteries. Atherosclerosis is a leading cause of cardiovascular disease. Traditional risk factors, example lipid levels and high blood pressure, don't fully explain early onset disease. Gut microbial metabolite, imidazole propionate, emerges as a non-traditional contributor to atherosclerosis. Imidazole propionate activates innate and adaptive immune responses via the imidazole 1 receptor signaling. The study investigators used mouse models, ApoE knockout mice and LDL receptor knockout mice. They were fed chow or high cholesterol diets plus antibiotics plus imidazole propionate for 8 to 12 weeks. In the human studies, two cohorts, the PESA cohort and the IGT cohort, the impaired glucose tolerance cohort. There were 400 in the PESA cohort and 1,084 in the IGT cohort, with subclinical atherosclerosis were evaluated using imaging. Techniques used: untargeted and targeted metabolomics, single-cell RNA sequencing, and plasmo and phosphoproteomics. Statistics: Spearman correlations, ANOVA, regression adjusted for age, LDL, and high sensitivity CRP, etc. What did they find? Imidazole propionate alone induced plaques in mice, p-value 0.001, independent of plasma cholesterol. In humans, higher imidazole propionate was associated with increased coronary calcium, p-value 0.0001. Arterial inflammation was confirmed with FDG uptake. Imidazole propionate levels strongly correlated with BMI, high sensitivity CRP, glucose, and visceral fat, p-value less than 0.01. Blockade of the imidazole propionate IVR axis with AGN 192-403 significantly reduced atherosclerosis, p-value 0.001. Imidazole propionate caused atherosclerosis through the imidazole 1 receptor, also known as nischirin, in myeloid cells. What was the limitations of the study? It was observational human data, causality inferred only from animal models, unknown microbial producers of imidazole, dietary correlation not fully understood as causal. Imidazole propionate is a gut-derived metabolite linked to early and active atherosclerosis, acts through the imidazole 1 receptor mTOR pathway in myeloid cells independent of cholesterol. Blocking the imidazole 1 receptor axis is a promising new therapeutic target. The accompanying News and Views article says what's new in this article, that is, imidazole propionate, a gut bacteria derived metabolite, promotes atherosclerosis independently of cholesterol levels. Imidazole propionate activates imidazole 1 receptor on myeloid cells, which stimulates the mTOR C1 pathway, which in turn boosts inflammatory immune cell recruitment and plaque buildup. Human and mouse models. Imidazole propionate levels were elevated in two human cohorts with subclinical disease and in atherosclerosis prone mice. An imidazole 1 receptor blocker, AGN 192-403, reversed the imidazole propionate's effects. What are the clinical implications? Imidazole propionate improves early detection when combined with traditional markers. Diets rich in fish, greens, fruits, and a tea are linked to lower imidazole propionate. Key takeaway: targeting the imidazole propionate mTOR C1 axis offers a promising cholesterol independent therapy for cardiovascular disease. Exciting stuff. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by Master MedFacts at mastermedfacts.com.